So today we're going to look at the structure of DNA, see what it's made up of, see how it replicates, and finally see how our cells can sort of read the DNA to determine the type of protein that's going to form. Basically to take the DNA code and translate it into what we'd call an amino acid sequence that forms proteins. Now the structure of DNA, if we were to look at it, an easy way to think about it is kind of like a spiral staircase. You've got sort of the side rungs or the rails that are along the side, and then you have these steps that are in between. Now if we look at a more molecular model type image of the DNA molecule, you have these repeating red, white, red, white, red, white that make up the sides, and then you have these balls in the middle here that pair up. And I don't know if you can see very well, but there's an orange one here that pairs with a yellow one, a blue one with a green one, and then here again it shows a yellow one with an orange one. So that's something we'll get into in a second, talking about the base pairs that form. Now, these molecules, these structures or chemical subunits we call monomers, are known as nucleotides. And that's what DNA is made up of, are these things called nucleotides. And a nucleotide is something made up of three parts. It has a phosphate molecule, it has a sugar molecule that we call a deoxyribose molecule. In Bio-30 we have deoxyribose molecules, and we also have ribose sugars. We're not going to go into that in Science 30. That's the difference between what makes something a DNA, deoxyribonucleic acid, and something called RNA, or ribonucleic acid. So we've got a phosphate molecule, a deoxyribose sugar, and then we've got 4 different nitrogen base molecules. And that's really what makes each nucleotide different. Each nucleotide, when we get into the 4 types in a second, each have the exact same sugar and phosphate, but it's these nitrogen bases that make it different, that make each nucleotide different. Now this would be a basic diagram out of your textbook of what a nucleotide would look like. You have a phosphate group, you have a sugar group, and then you have a nitrogen base. If we wanted to get into the more detailed chemical structure of it, there's the phosphate again with the phosphorus. Here's your sugar molecule, your 1, 2, 3, 4, 5 carbon sugar molecule, and then there's your nitrogen base that comes off of that sugar molecule. Now the sugars and the phosphates make up the backbone of the DNA. If we were to look at a fairly simple structure of a DNA molecule, here you can see how it's all wound up, it's all coiled and twisted, and then if we were to separate the 2 strands of DNA, you would see this phosphate sugar, phosphate sugar, phosphate sugar, that keeps repeating, going down this side rung, the blue part on this diagram. Okay, so constant sugar, phosphate, sugar, phosphate. And the other DNA molecule, the other strand, of the DNA molecule, going in the other direction, again, that's going to be something that's the Bio-30s look at with 5 primes and 3 primes. If you've taken Bio-30, that should be familiar to you. But again, it's a sugar and a phosphate that repeats. And it's in the middle here that we have those nitrogen bases. So a nucleotide, again, would be this right here. That would be one of the nucleotides. Let's flip over, I'll make a little sketch here. That right there would be a nucleotide, okay, where you've got your phosphate, you've got your sugar, and you've got your nitrogen base. Now, if we look at the middle here, we wind up having some pairs that keep happening. We get a T going with an A going with a T, a C going with a G, and a G going with a C. And what those letters stand for, those A's, C's, T's, and G's, those are the nitrogen bases. So the adenine, or A, the C for cytosine, the T for thymine, the G for guanine. Again, don't need to worry about memorizing those names. Those names are given to you in your data book. Now, as you saw from that last diagram, we saw some pairs that happened. We just saw that the A always went with the T, and the C always went with the G. So if we look at this diagram, you can see that the A has a shape that complements the T, the C has a shape that complements the G. And if we were to go back to this diagram, it's not necessarily that the shape itself complements, it's because there's a certain number of bonds or bonding sites between each. So an A, for example, an adenine, and a T for thymine both have 2 bonding sites, whereas a C and a G both have 3 bonding sites. So a T could never go with a G under normal conditions, and we'll talk about mutations later, but normally it wouldn't be able to bond with each other because a T has 2 bonds and a G has 3 bonds. So they wouldn't be able to match up properly with each other. And just like an A wouldn't be able to go with a C because they have a different number of bonding or attachment sites. So what you will need to remember in Science 30, obviously what a nucleotide is made up of, but that a C and a G pair and an A and a T wind up always pairing with one another. So that's what this next point here was talking about in your PowerPoint. These are called base pairs or base pair rules. So something that we could do is if we know that, let's say we were doing a DNA sample and we wanted to figure out, I don't know, we wanted to figure out what the percentage was of the different nucleotides. So if we have A's and T's and C's and G's, and let's say they told us in this analysis that let's say 13% of our DNA sample was made up of adenines. Then what we should be able to figure out from that is, since A's and T's both pair with each other, that if there's 13% adenines, there also has to be 13% thymines. Okay? Now what about these other two? How do we figure out the amount of cytosines and guanines? Well, this has to add up to 100. Okay? So if we've already got 26% right there, 100 minus the 26 is what, 74? So we've got 74% left over. That 74% needs to be split evenly between these two because cytosine and guanine represent the same amount. So 74 divided by 2, what, 35, 37. So we get 37% of our DNA sample made up of cytosine and 37% of our sample made up of guanine. So kind of an interesting little way for us to take some of our information that we know about the base pair rules and then apply it to something like this that maybe shows up on a diploma exam. Now the DNA molecule is actually something that's only recently, if we think of science as a long history of invention and discovery in science, relatively recent discovery. Relatively recent for you guys is old, but 1950s, 1953, Watson and Crick, 2 guys who discovered the DNA molecule. Really interesting story about these guys. I would do a quick little YouTube video clip on, or search on Watson and Crick and a lady named Rosalind Franklin. The ladies in the class will be quite disturbed by how Rosalind Franklin's contribution was totally put to the side by these guys, and her contributions were not included when they published their report about what the DNA molecule looked like. So something interesting for you to check out with the science and the history of the DNA molecule. If we were to get a sample of DNA on a test like this, so if we had to get the, let's say we had the code AAT, if you're reading right here, AAA, TGT, CGC, C, and T, and on a diploma exam, maybe they would ask you, what is the complementary strand of DNA going to look like for this code? You need to remember that A's go with T's and C's go with G's. So if that was what the code was on one strand of the DNA molecule, A, etc., I'm going to write just over top, actually right at the bottom here, the complementary strand. So if you had an A, that's going to pair with a T. The A there going to pair with another T, and so on. An A on that strand with a T. Now we get a T, so a T is going to pair with an A. A G is going to pair with a C. The T there with an A, and so on. C with a G, and so on for the rest of that strand of DNA. So that would be a familiar or a similar type of question on your diploma exam. Now moving on to this next page here, we need to review the process for making a new strand of DNA in this process called DNA replication. Remember, every time a cell divides, it needs to make a copy, an exact copy or nearly exact copy of what the DNA molecule looks like. That's important so that each one of the daughter cells will have the same instructions, the same genetic information as the original parent cell. Now what this animation is going to show is a little bit complex. It shows how this DNA molecule is going to be replicated. Now there's going to be some terminology in here, helicase, DNA polymerase, etc., 5' and 3'. And as mentioned earlier, there's a lot of material here that is covered in Bio 30 that is not the same level as what we get into in Science 30. So look at sort of the overall view of how this is being replicated, and I'll sort of walk you through as we go along. Notice the first thing that's happening. The DNA molecule is being unzipped. Okay, so the strands are being separated. Okay, now what's going to wind up happening next is we're going to need to make copies. So notice there's an open space right here. This would be where it's sort of unzipped. An adenine needs to pair up with a thymine. So this thymine, you'll be able to follow this molecule moving along, and we'll wind up forming some new DNA. So just watching along in this part here, you'll see new red. So this is new DNA that's getting formed. And then the other strand of DNA, this one down here that's separated from this one, has to get made in the opposite direction. So again, it has to do with the way DNA gets copied, and then these sections have to get clipped together or sort of glued together. There's a little primer so the polymerase molecule knows where to start manufacturing. And then you notice it'll get, watch, there's a little gap right there, and it gets glued together. So we wind up making two identical strands of DNA. So what did we see happen? We saw the two strands of DNA molecule unzip and uncoil. It went from a coiled up double helix shape to two separate strands of DNA. Then what happened was some free-floating nucleotides, some free A's, T's, C's, and G's were brought in, and they were attached to their complementary strands or complementary nucleotides. And then finally, at the end, the two new strands of DNA zip back up, they recoil, and now the cell is ready to divide. So a couple images here from your textbook showcasing this. This is something that would be good to watch a few times on the YouTube clips. Again, knowing though that the YouTube clips, a lot of them are a lot more detailed than what we need to go into for Science 30. Now the whole point of this at the end of the day is to make protein. That's what DNA is. It's, like we said earlier, a big cookbook, instructions for making proteins. Now proteins are things that are made up of smaller subunits called amino acids. And if we were to look at this idea, we would have maybe a whole bunch of little circles. And these circles here would represent individual amino acids. Okay, 20 different amino acids. And they all get linked together. I'll call this a peptide bond, links individual amino acids together. So this right here, this guy, is an amino acid. Let's put an A amino acid. This entire thing, that whole thing there, is called a protein. Tough to write with a mouse. Is a protein. So a protein, big long chain, could be 20, 30 amino acids long, could be hundreds of amino acids long. But what makes a protein have certain characteristics is the amino acids that it's made up of. So the analogy I use in class, I talk about a protein kind of like a necklace, a pearl necklace, okay, made up of a whole bunch of pearls or maybe even beads that are different colors. So you might have 30, 40 beads that are on your necklace. The necklace would be a protein, okay? Each of the beads that is of a different color would be like an amino acid, okay? Now, if we move on here to the next page, how do we read the DNA sequence? We learned years ago that the DNA code is read in triplets. It's read in 3-letter sequences. So if I had the code A, C, T, G, A, C, A, T, for example. And it continued on that way, and it continued on that way. What we do is we take the DNA sequence and we split it up into 3-letter chunks. And then what we're going to be able to do is go to a page that's in our data books and in our textbook, and the cells know how to read this code. They know what AAC stands for, TGA stands for, and so on. Each one of these sequences stands for a certain amino acid. So let's go to the next page here, and I'll show you what these codes are. This is not something you will have to memorize. This is something that is in your data book. It's something that you're able to use on an exam, use in a quiz, and you'll be able to read the instructions, read a DNA sequence, and determine what the amino acid sequence is. Now, key to point out, Bio 30 people, the way we're going to do it here is going to be quite different than the way you're going to do it in Bio 30. So really important that when you're taking your Bio 30 class, you're not applying the rules here straight to what you'll do in Bio 30. You that have taken Bio 30, you might remember that there were these things called uracils, U's, that are in place of T's. You had to learn about mRNA. There was a whole bunch of, whole slew of extra steps that you would have had to do, and we don't do those here. So for example, again, let's just take a different DNA code. Let's say we're going TGA, CA, T, TAC. Okay? All these codes are listed in this page. You can start with the first base, the first nucleotide of that triplet code, we call a codon, and you'll be able to find it, and then you'll be able to match it up with a particular amino acid. So this right here, for example, CTG, codes for the amino acid leucine. Now look, there's a whole bunch of them that are coded for, like leucine is coded for by more than one sequence. So that's important. There are a lot of these amino acids that are coded for by multiple sequences. We're not all exactly the same genetically, but we're still able to produce the same proteins, like insulin, let's say. Insulin in me is going to have the same amino acid sequence as insulin in you, but I'm not going to have the exact same DNA sequence to produce insulin as you would. So for example, for me, let's say one of the amino acids in the insulin molecule is serine. Okay? My code might be TCT in my DNA molecule for that section, but in you, it might be TCC. So we're different genetically. We produce a different or have a different DNA code, but protein-wise, we wind up producing exactly the same amino acid. Now, there are a couple other ones that are in here that are useful. These guys here, TAA, TAG, and TGA, notice they say stop. You can probably imagine what this is. Right? At the end of each gene, there needs to be almost like a period or an exclamation point or some kind of thing at the end of a sentence that marks the end. So each gene has the same thing. It will have a series of bases, TAA, TAG, or TGA, that marks the end of the gene. At the beginning of every gene, you'll also see a start code, so kind of like a capital letter at the beginning of a sentence. ATG, or methionine it's called, is a start code. So if I'm reading along, here I've got my DNA sequence. Remember we said we'd want to break our DNA sequence up into triplets. So we'll physically draw some lines. I would do that on my exam. I would draw lines. I would maybe underline, whatever works for you so that you can see that there are 3 different codes. And then I look TGA. So what I do is I just look for the first letter, T, and then there's second base, G. So you know it's going to be in here somewhere. I just look for the first letter and then I skim across. Now, maybe this was a bad one for us to start with because TGA stands for a stop code. So that would be bad. If this was the middle of a gene and we had a stop code in the middle of the gene, that would be like you trying to, I don't know, open your locker combination and only doing the second number in your locker combination and trying to open your locker. It's not going to open. Same thing with a protein. If you don't have the right sequence of amino acids, then the protein isn't going to function properly. But we're just trying to illustrate a point of what this code stands for and how you can figure out the amino acids. CAT, so again, I look for the first base, I go across. CAT, right there, stands for the amino acid histidine. And TAC, I come up again, T, I go across, TAC, right there, stands for the amino acid tyrosine. Okay? So that's one of the things that we're doing. You're going to get a DNA sequence, and you'll have to figure out the amino acid sequence. The other thing that we could do is give you the amino acid sequence. So we can write serine, I'll just put serine, oops, serine, and then let's go glycine. Let's put the first 3 letters for these, and then let's go alanine. So let's say they gave you this was your amino acid sequence, just a section of your amino acid sequence, and then you needed to find out what the DNA sequence was going to be. So what I would do is I've got serine. So here's serine. Whoops, here's serine here. And I've also got serine over here. So be careful because it's sometimes found in different places. So I could have the code TCT, TCC, TCA, TCG, AGT, or AGC as my first 3 letters of my DNA sequence. Then glycine. Glycine, right here, glycine. I've got GGT, GGA, GGC, GGG. Any of those letters could be the next 3 letters in my sequence. And then finally alanine, alanine, right here. I could have the code GCT, GCC, GCA, and GCG as the next part of my sequence. So in your textbook, there is a section where you can get some practice with this. It's right here, where you're going to take your DNA sequence and you're going to turn it into amino acid sequence, and vice versa. You're going to take your amino acid sequence and convert into your DNA sequence. So on page 114 in your textbook, that will give you some good practice doing this part.